Yeah, so Thomas, thanks a lot for the introduction. And it's really a very special honor for me to speak at the occasion of Werner Reichardt's 100th birthday, because the focus of my research for the past 35 to 40 years has been to find the neural implementation of Werner Reichardt's motion detector model in the fly visual system. So my talk will take you, from the original model to the neural implementation in the fly visual system. And let me start by introducing the fly visual system to you a bit in more detail. Here you see a fly with its nervous system in blue. And if we zoom into the head ganglion, the brain of the fly, then you see that large parts of the neuropil are taken up by the two red structures here, which are the optic lobes. And the optic lobes in the fly are like the mammalian retina, just concerned with image processing, that the signals they receive from the facet eye. And the optic lobe is built from several layers of neuropils called the lamina, the medulla, the lobula, and the lobular plate. And each is built from in a retinotopically arranged columns, which sort of repeat the columnar structure of the retina. And now here's the central phenomena that we want to understand. If you move a bar from the left to the right and back again, and you record from one of the photoreceptors, then you see the same signal each time. Photoreceptor just depolarizes, and by looking at these signals, you cannot tell the direction of the image motion. So it's a non-directional signal. Now you take this electrode and stick it into one of these lobular plate tangential cells, which pool the output signals of many hundreds of local motion sensitive neurons in the fly optic lobe. Then all of a sudden, you see a direction selective signal. The cell depolarizes in response to rightward motion. That's called the preferred direction. And they hyperpolarize in response to leftward motion. That's called the null direction of the cell. So within a few synapses, you go from a non-directional to a directional selective signal. And how come? Well, that's exactly what Werner Reichardt and Bernhard Hassenstein's model predicted. They did behavioral experiments, as we already heard, on this beetle working on the spherical Y maze. And from these behavioral experiments, concluded such a motion detector model, which they published in '56, which is just a year before I was born. And this is the model with all sort of the bells and whistles, with all the different filters, sort of hard to interpret at first sight. But this is the model in its essence. And this model consists of two mirror-symmetrical subunits, one in blue here, which is tuned to rightward motion, and one in red, which is tuned to leftward motion. And within each of these subunits, the signal from one photoreceptor is delayed by a low-pass filter and subsequently multiplied with the instantaneous signal from the neighboring photoreceptor. And if you look at the output of these multipliers, you see all of a sudden directional selective signals. So this one, has a strong response to rightward motion and a small one to leftward motion. And the other one, it's the other way around, has a small response to rightward motion, a large response to leftward motion. And when you now subtract the output of these two multipliers, then you see a fully opponent signal, as we have seen before, at the level of the tangential cells. So when I arrived here in Tübingen as a young postdoc in '84, there was already a lot of evidence in favor of this model in the fly visual system. But all these predictions and experiments concerned the steady state response of the motion detector, and no one had looked really at the dynamics of this model. And so together with Martin Egelhaf, who was a grad student with Werner Reichardt, I was a postdoc with Kai Götz, and Martin had just finished his PhD thesis, so he also became sort of more independent. we teamed up and we sort of thought about, testing this model in more detail, and in particular in the dynamic regime. So the first thing that we did was that we simulated an array of Reichardt motion detectors and looked at the responses of this motion detector array when the pattern starts moving. And what we saw was that there was initial ringing before settling to steady state. And this ringing was exactly at the temporal frequency of this grating motion. And so this was very sort of unusual. So we did experiments and recorded from the large tangential cells in the blowfly, and we indeed saw this ringing at exactly the temporal frequency of this grating motion. And so that was sort of very rewarding and convinced us that, okay, this model must be true. And so then Martin and I came up with another idea, and we wanted to look now not at the array of motion detectors, but what would the signals of a single motion detector look like in response to grating motion. And this is what it was. So in response to preferred direction motion, this computer simulation of a single motion detector predicted an ongoing modulation with exactly the temporal frequency of the grating for preferred direction, for null direction. But we saw something else, and that was the second harmonic, which comes about by the multiplication of two sine waves. And that was very intriguing. So we wondered whether we could somehow record such local signals from a spatially integrating neuron, which is the tangential cell. And then together with Werner Reichardt, we came up with the so-called slit experiments, where we prevented spatial integration by presenting the grating just through a slit to the fly. And so we would have all those local motion signals sort of that are stimulated in synchrony, but we can record it from the large integrating tangential cell. And this is what the signals look like that we recorded. And we thought, this is beautiful, very much in line with our computer simulations here. And again, we realized the second harmonic in the response. And so a further proof of Werner Reichardt's motion detector model in the fly in very much detail, I would say. So these experiments sort of not only convinced us about the truth of the model, they also allowed us to pass the habilitation, which is the sort of an exam that you had to take in the German academic system before you can apply for a professor position. And so these pictures were taken from our habilitation party. So this is Martin Egelhaf. This is Werner Reichardt. I have no idea who this, young chap is. Honestly, it's me. No one recognizes me, so I have to tell you. This is Martin Heisenberg, peeking over Martin's shoulder. And then another picture. This is Martin. This is me. This is Karl Götz. And Karl just congratulates us to this kind of bacon stuff. And, and that's a bacon version of the Reichardt detector that was baken by Johannes, who's here in the audience. Johannes, yeah, raise your hand. So he's responsible for that. And so we went from testing the model to tasting the model. And that's what Johannes did for us. Because honestly, we were, Martin Egelhaf and I were really, not only excited by the Reichardt model, we were, obsessed somehow. And and so Johannes, gave us this present, and it really convinced us that Reichardt detector also tastes well. It's a great model. So. Here, yeah, on the left side, that's the other, there were two versions that Johannes did for us. And the left and the right half detector were, pictured as Martin and me. Johannes, thanks again. Anyway, so after sort of accumulating so much of experimental evidence for the, for this model that it describes so well the process going on between the fly photoreceptors and the lobular plate tangential cells, the natural question was, which neurons do this computation that is defined by this model? And so looking at all the different columnar cell types in the fly optic lobe, you see this jungle. That's all the different, about 100 different cell types that are found and have been stained by Fischbach and Dittrich, published in '89. And each of these cells is one cell type. It's shown once here, but it lives, 750 times in the optic lobe. And now this was very sort of disappointing because it's a catalog of sort of the silent cells, because the response properties of none of these cells were known at the time. No one has recorded from any of them, at that time. And so, on the other hand, we had, this model which describes the process going on between photoreceptors and tangential cells so well. And we were, somewhere in there, these neurons doing these computations had to be, but how can you find them? And at that time, this model was really just a black box. And I already told you that all of us interested in physiology at the time worked with blowflies, because we could at least record from the tangential cells with sharp electrodes. Now, Drosophila at that time was not a real alternative, because we didn't have an expression system like the GAL4 UAS, so we could not target any protein to any cell type. We didn't have genetically encoded calcium indicators. So, Miyawaki et al., the paper, from Roger Ten's lab came out in '97. Patch recordings was not established yet in the fly. And not even in '89, the two-photon microscope was born. So with any regular laser scanning microscope, you would just blind the fly if so nothing was there in place. But I was coming from a Drosophila lab. I did my PhD with Martin Heisenberg, who worked with Drosophila. And so I was sort of keeping an eye on the development of the Drosophila field. And I had to wait. And in the 2000s, all these techniques came up and then sort of were in place. And this is when we shifted from the blowfly to Drosophila. And really, Drosophila, it paid this move. And it helped us open the black box. And, we made a number of discoveries. First thing was that we found that motion is detected in the fly visual system in two parallel on-off systems, just as had been suggested by Nicola Franceschini before. Second thing was that we found the neurons that are equivalent to these multipliers are the T4 and T5 cells in the fly visual system. Then we found that this subtraction stage is implemented by LPI interneurons in the lobular plate. And just 2 years ago, we found the interesting biophysical implementation of the multiplication in the on pathway on the dendrites of the T4 cells. And for the next 20 minutes or so, I will take you, first introduce the T4, T5 neurons to you. Then I will tell you about the biophysics of this nonlinear interaction here. And then finally, I will tell you about the implementation of the subtraction stage of the motion detector. So what about T4, T5 cell? Like any other neuron on Earth, in any animal species, it has first been discovered by Ramon y Cajal, right? That's the rule. Yeah? And he published in 1915 a book together with student Sanchez. And if you look at the different pages here, you see all of a sudden this neuron, which has its dendrite in the medulla and an axon terminal in the lobular plate, the soma here. And this is a T4 cell. And a T5 cell has the dendrite here in the lobula, axon terminal also in the lobular plate, and the soma also here behind, posterior to the lobular plate. Now, meanwhile, we know that T4 and T5 cells consist of 4 distinct subtypes, which all have the dendrites intermingled here in the respective layers, but send their axon terminals into one of the 4 distinct layers of the lobular plate. And so a couple of years later, we got a driver line from Gerald Rubin and Alyosha Nern from Janelia Farm, which was specifically targeting the T4 and T5 cells. Here you see the dendrites of the T4 cell under the confocal. Here, the dendrites of the T5 cells. And here you see the axon terminals in the 4 layers of the lobular plate. And here, Matt Majzek was a grad student in the lab, and Jürgen Haag is a long-term collaborator of mine who's also here. Jürgen, where are you? They took these flies under the confocal, under the 2-photon microscope, and this is how this area looks under the 2-photon microscope. And you see anatomically already the 4 layers, 1, 2, 3, 4 of the lobular plate. Now, if Matt and Jürgen stimulated these fly visually with grading to the right, they saw a very strong activity in layer 1. If they moved the grading from right to left, they saw a strong activity of the calcium indicator in layer 2. They moved upward, they saw activity in layer 3, downward in layer 4. If we now take such a picture and label each pixel with the color of the motion that it responded the most, we get these 4 beautiful layers of the lobular plate, which says that the 4 subtypes are distinct from each other by their preferred direction, and they follow the 4 cardinal directions of motion, despite the fact that you start off with an hexagonal symmetry and geometry in the eye. Now, this says something about the T4 and the T5 cells, but what is the difference between the T4 and the T5 cells? For that, we had to switch from grading motion to edge motion and presenting the bright and dark edges. And we also had to have driver lines specific for T4 and for T5 cells, which again, we got from our colleagues at Janelia Farm. And what we saw was that T4 cells preferentially respond to bright edges, to on edges, and very little to dark edges. And the opposite is true for the T5 cells, which respond very little to bright edges, but very strongly to dark edges. So these are the response properties of the T4 and T5 cells. And after discovering them as the primary motion sensing neuron in the fly visual system, the next question was, how do they become direction selective? How do they compute the direction of motion? Well, there are 2 alternatives. Alternative 1 is the one that I already described to you. You take the signal from one side, you delay it by a low-pass filter, and then you multiply it with the non-delayed signal in the center. That mechanism is called preferred direction enhancement and was the proposal of Werner Reichardt and Bernhard Hasselstein. As an alternative, Barlow and Levick had proposed that you take the signal on the other side of the motion direction, pass it through a low-pass filter, and now block this signal. And Tommy has already alluded to that. That's called null direction suppression, and a division was proposed for that. So now, in order to decide between preferred direction enhancement and null direction suppression, Jürgen Haag, again, did the following experiments. He used a telescope to place visual spots exactly on the hexagonal array of the fly eye, used the calcium indicator to record from T4 and T5 cells that have their receptive field exactly there. And now he did the following. He first stimulated each one in isolation. These are those traces. And then as a sequence. And then he compared the response to the sequence with the sum of the individual responses to extract the nonlinear component. And if he does that along the preferred direction of the cell, he sees a preferred direction enhancement. The nonlinear component is positive, right? So that clearly speaks for Werner Reichardt and Bernard Hasselstein's idea about preferred direction enhancement. But when Jürgen inverted the sequence, moved the dots from the right to the left, then he saw a clear sign of null direction suppression. Now he has a negative nonlinear component when comparing the actual signal or response to the sequence with the sum of the responses. So what we're dealing here in the fly is not one or the other, but in fact, both processes, together contribute to the direction selectivity of the T4 and the T5 cells. And that was then also later confirmed by a paper from Tom Clendenin's lab, actually at the same time, not later. So we all, agreed on this tripartite input structure of the T4 and T5 cells. Question is, of course, now, which cells are providing the input? That's still sort of a formal model. Unfortunately for us, our colleagues at Genelia Farm not only provided us with these driver lines, they also made the fly connectome and figured out the connectivity between all the different neurons in the fly optic lobe. And so they could tell us that T4 cells, receive input from a total of 7 different cells. And I'm just concentrating here on the major inputs. And the major inputs to the T4 cells are called MI9, MI1, and MI4. And for the James Bond fans, you have the question, is MI6 here? No, it's not. Yeah, so MI6 is not there. But what was really rewarding about these anatomical results was that it sort of, again, reflects this tripartite structure that we found and postulated from our functional experiments. And it's reflected in the anatomy. And the T4 cell tuned to rightward motion, has the MI9 cell here, MI4 there, and the T4 cell, for the opposite direction, then has MI9 here and MI4 there, right? So a couple of students in the lab and postdocs then did calcium imaging to study the receptive field properties of these cells, and they used white noise motion and then did reverse correlation to extract the spatial temporal receptive fields and then fitted linear filters to it. And this is what they look like. Most cells have rather small receptive field of about 6-degree half-width, with an antagonistic surround. Interestingly, as expected for a T4 cell in the on pathway, they have on centers with the slight off surround, but MI9 is odd, right? It is an off cell, has an off center with an on surround, yet it belongs to the on pathway. Now, looking at the temporal properties, I show you the step responses of these fitted filters, and we see very much in line with our expectation, the flanking cells are low-pass filters, that's a step response of a low-pass filter, whereas the central one is more band-passy and faster, right? So that fits, and we can account for the receptive field properties and directional tuning of T4 cells by just taking, the inverted signal of MI9, multiply it with MI1, and then divide by MI4, and we get nice directional tuning, as we've seen in the experiments. But, this is still, multiplication, division. We wanted to know, the biophysics of these computations. I wanted to have an electrical equivalent circuits based on conductances, which then do these computations. And as a first step towards such an understanding, you need to know the transmitters. And fortunately, we and others, figured out that MI9 is glutamatergic, this one is cholinergic, this one is GABAergic. Well, now in the transcriptomic age, you can also look at what receptors T4 cells express. And we found nicotinic acetylcholine receptor subunits. We found GABA A receptor subunits. That's all sort of as expected. Unexpectedly was to find a high expression level of glucl alpha receptor. Now, glucl alpha is a glutamate-gated chloride conductance, which makes glutamate an inhibitory transmitter in this case. Now, I was very happy to see that because I thought, that takes care of the sign inversion, right? So, somehow it has to do that. And but before, coming to functional considerations, we also looked now at the distribution of these different receptors on the dendrites of the T4 cells in order to see whether they match this input structure, right? And this was work that was done by two students in the lab, Sandra Fendel and René Vieira. And this is how the dendrites look at the EM level of the different subtypes of T4A, B, C, and D level. And that's from a paper from Shinomiya and colleagues from Genelia Farm again. What you notice is that, you can already tell by the anatomy of the dendrite what its preferred direction is, because the dendrite is always oriented against the direction of motion it responds to. So this one has the tips to the left, it responds to rightward motion. This one has the tips to the right, responds to leftward motion. This one has the tips downward, responds to upward motion. This one has the tips upwards, responds to downward motion, right? So you can tell from the anatomy already which subtype that is. So now Sandra and René developed a so-called flip tag technique to label the receptor proteins and then localize them on the dendrites of an individual cell. And this shows one example of a T4A cell, which has the tips to the left. And you see glucl alpha here at the tips, where MI9 is providing the glutamatergic input. You see the nicotinic receptor spread all over, so to say. I will quantify that later. And GABA A here primarily strongest at the base. And now to quantify all that, they did this, with all the different subtypes and oriented such that the tips are to the left and the base is to the right. And what you can see is that glucl alpha is at the tip, the nicotinic receptors are in the center, and the GABA receptors are at the base of the dendrite. And this again reflects this tripartite structure that I've showed to you before. So now about functional consequences of all of that. So this is how it could work. And so in the dark, MI9 is active. In the dark, MI4 is 0, is not active. And now we have a flash of response to the center, MI1, it will respond like this. Now, due to the activity of MI9 in the dark, this one keeps all the time these chloride channels open, and that will reduce the input resistance. So you have a low input resistance, which will further go down when MI1 becomes active. And the membrane potential is sort of this depolarization. That's a computer simulation, just sort of in principle, right? So now what happens if I move an edge from left to right? This bright edge will inhibit MI9, right? And this inhibition of MI9 will lead to closure of the chloride channels, which will lead to an increase of the input resistance. And now activation of MI1 leads to a larger depolarization because you have a larger input resistance, right? That is the idea. And for the opposite direction, the two signals of MI9 and MI4 overlap. So you have a further decrease of input resistance. And consequently, you have a smaller response than to the MI1 signal alone in isolation, right? So this is how it could work. And it was not the first one to express this idea. Tommy and Vincent Torre had this idea before, actually long before. In '78, they published this paper. Tommy showed it already in his talk. And here, you see the idea that, with basically shunting inhibition, you have the nonlinearity required for null direction suppression. And the idea that I presented to you here is simply turning that around and propose release from shunting inhibition as a mechanism to have the positive nonlinearity for preferred direction enhancement. And now, in order to figure out how in reality it really does work, we had to overcome one major problem. And this major problem was that all responses we had until then were from calcium imaging. But with calcium imaging, you could not measure any ionic currents. You could not measure input resistance or anything like that. You wouldn't also see hyperpolarization, whatever. So there was one paper from Michael Reiser's group coming out at that time. Eyal Grundmann and Michael Reiser from Genelia Farm published their first wholesale patch recording from the somata of T4 and T5 cells. Now, this was an incredible achievement because the somata are 2.6 micrometer diameter. So it was amongst the smallest cells you can get. But they managed to do it, so we thought maybe we can manage also to record from them. And then came this brave postdoc, Lukas Groschner, came into the lab and said, oh, I can do it. And within a few months, he could regularly record from these T4 cells. And he also taught a grad student, Jonathan Malis, to record from the different input neurons. And they used the same stimulus set. So we knew the input signals, we knew the output signals, and then we could really check what's going on there. And this is how it does work. So on edge motion to the right, you see a strong suppression of MI9, followed by a rise of the signal in MI1, which then is followed by the rise of the signal in MI4. But those two inhibitory neurons, MI9 and MI4, leave open this window of opportunity, so to say, for the excitation to come in. And consequently, you see a large depolarization in the T4 cell, right? And for the opposite direction, the two inhibitory signals overlap. There's no such window of opportunity. Consequently, you see only a small depolarization to null direction motion. Now the question is, that all fits and makes sense, but there are a number of critical questions you can ask. The first question is, is glutamate really inhibitory? I mean, there's a lot of circumstantial evidence, but who knows? So Lucas puffed glutamate onto the dendrite of the T4 cells, and he saw glutamate really hyperpolarizes these cells. If we block expression of the glucial alpha receptor, we don't see a response. We see a depolarized sort of resting potential in the T4 cells. Next question is, does glutamate really control the input resistance of these cells? And answer is yes, it does. Here you see the responses to a series of current injections into T4 cells. You see that a large amplitude responds to depolarization and hyperpolarization. If you puff glutamate just before these current injections, you see that the response amplitude is much reduced. So it's half of what it is before. Next question is, now this is artificial application of the transmitter. Does the change of input resistance really happen during visual stimulation? So Lucas repeated these experiments, measured input resistance and membrane potential at the same time. And what he saw for a bright edge moving in the preferred direction, you see the nice depolarization here, and the dashed line represents the input resistance, and it rises from about 5 gigohm to 8 gigohm just before the cell depolarizes. So we do, in fact, see this change of input resistance in visual motion. The last question is, how does the directional tuning now change in the T4 cells with and without this glucial alpha receptor? That's the polar plot of the directional tuning of a healthy T4 cell before Lucas, Jonathan, and Birte take out the glucial alpha receptor by RNAi. and you see it's way more roundish. So it really contributes to the directional tuning of the T4 and the T5 cells. And so now, knowing all the signals, we could build a T4 cell model using these measured input signals based on the conductances and everything. And these are the experimental data for preferred direction, null direction at 15 degrees per second. And this is what the model response looks like. The same thing for 30 degrees. Again, this is what the model looks like. And now for a full data set for gradings moving at 5 different velocities, on-edge motion, off-edge motion. In blue is the experimental data for preferred, in null direction in red. These are the experimental data, and this is what the model does. And I think, we capture it in pretty much detail. So now that we could record, from these T4 cells, it was very interesting to me to compare that with, our early predictions that we did with Martin Egelhoff back in the 80s and with Werner Reichardt here. So this is now the paper again that I showed to you in the beginning. These were the early predictions. This computer simulation. This is the the recording from the tangential cells. The slit experiment, and these are, the T4 cells responses that Lukas Groschner recorded, and I, find that amazingly similar to what we've, did, 30 years before, and including all these sort of second harmonics that we see here, which are indicative of the multiplication. So I would say pretty good job. So let me just summarize before we, do the last 5 minutes, and then can have lunch. So the looking for, the elementary motion sensing neurons in the fly optic lobe, we found the T4, T5 cells being responsible for that. And these form these beautiful functional layers representing the 4 directions of motion in the lobular plate. And about the mechanism, we found that if we compare it with the original model, and now, simply take or concentrate on the subunit here, that it's not just 2 inputs that are provided here, but 3 spatially distinct inputs and a combined action of preferred direction enhancement and null direction suppression. And with respect to the biophysics for preferred direction and null direction suppression, we're pretty much done at T4 cells, and we're still working on it in T5 cells. But, that's very rewarding. But what about this subtraction stage? That was something that I promised in the beginning, but didn't talk about so far. Well, if we compare now the preferred directions of the HS and VS cells with the ones of the T4, T5 cells, you will see the following. These are cells that Lisa already introduced to you. They depolarize HS cell, to preferred direction, rightward motion. They hyperpolarize for leftward motion. These ones depolarize downward motion, hyperpolarize for upward motion. If we take a look at this lobular plate cell from the top, we see that the HS cell dendrites have their dendrites in the first layer, where T4, T5 cells have their terminals that respond to rightward motion. VS cells have their dendrites in the 4th layer, where the T4, T5 cells terminate that respond to downward motion. So this matches, right? It fits. It fits for the preferred direction, but who's causing this hyperpolarization? And so if we go, to such a scheme where you have the 4 layers of the lobular plate and your 4 sorts of local motion detectors, already, with Martin Egelhoff, we concluded that the subtraction stage is implemented on the dendrites of the tangential cells. Now, if you go through that, just look at this little scheme. This is the T4 cell, this is T4B cell, this is T4C cell, and this is now T4D cell, right? But what about these inhibitory, neurons that we postulate somehow? So does each of these T4, T5 cells have an inhibitory twin? the answer is no. We calculated them, we counted them, and the numbers really fit. We have 8 of these cells per column, 44 cells, 45 cells. They're all cholinergic. There are no inhibitory T45 cells. So what is going on? Well, that's the story of Alex Maus, a postdoc in the lab at this time. And he applied optogenetics to the fly visual system. And here you see the T45 cells expressing channel rhodopsin, and he recorded from the VS cell. And when he stimulated the T45 cells optogenetically, he saw the following. A 2 millisecond pulse of light causes the tangential cells to depolarize rapidly, and followed by a delayed inhibition. Right? So somehow we thought, okay, the excitation is provided by the T45 cells monosynaptically. They synapse onto these cells. Meanwhile, we know that this is the case. But there's an inhibitory interneuron in between. So this one is not a direct inhibition, it's a delay. So now we had a sort of search profile for this inhibitory interneuron. And we postulated that it should be B-stratified, 2 layers of the lobular plate it should cover. It should be postsynaptic in 1 layer, and then presynaptic in the adjacent layer. And I'm really not making this up. We really had, these data. We postulated these neurons, but no one had seen such neurons before. So I again send an email to Janelia from Alyosha Nairn. He knows the anatomy like no one else. And Alyosha looked through all, catalogs of neurons, came up with this. Wow. We thought, this is amazing. Now, if you take these, lobular plate interneurons, if you, look at them from the side, you see the following. This is the lobular plate from the side. See the 4 layers formed by the axon terminals of T45 cells. And that's one of the lobular plate interneuron 3-4. It only goes into layer 3 and into layer 4. And if we, stain it for synaptotagmin, which is a presynaptic marker, you see it is only presynaptic in layer 4. So it matches the search profile perfectly. And so Alex and Katharina and Alexander in the lab then did calcium imaging and found that this neuron indeed responds to upward motion, as it should if it receives the input in the 3rd layer, right? Now, Alex then stimulated these interneurons directly, optogenetically, and what he saw was a monosynaptic fast hyperpolarization. So these really are inhibitory interneurons. We meanwhile know they are using glutamate as a transmitter, and the gluCl alpha receptor is expressed on the dendrites of the tangential cells. Now, the last question was, of course, are they really responsible for the null direction hyperpolarization of the tangential cells? And here you see preferred and null direction of the 2 parental controls, right? Nice and strong. If we block these LPI 3-4, the depolarization is untouched, but the hyperpolarization is completely gone. You rarely get such clean data. That's really amazing. And now there's a last point before lunch, just one last point. So what is the function of this lobular plate interneurons? In the old days, we thought the subtraction stage of the Reichardt detector is to improve and sharpen the directional tuning of the subunits. But the T45 cells are already highly directionally tuned. So what is the function of that? Here you see a VS cell which has a homogeneous optical flow field preference which is downward. Now, what would such a neuron respond to say, a random optic flow or an expansion optic flow or a looming square? Now, you have a number of motion vectors which stimulate this cell, but you have an equal number which inhibit this cell. And so as a net, when we do a computer simulation of the full circuit, including these inhibitory neurons, then in the computer simulations, the response to these other flow fields, non-preferred flow fields, so to say, is pretty much zero. Now, if we take out this inhibition from these LPI cells in the computer simulation, now all of a sudden, then you only have the excitatory input and you see, a large depolarization to the random flow fields, to the expansion flow field, to the looming square. And now Alex then did these experiments. First, he recorded from intact flies with everything intact. You see very little response to these different flow fields. But when we block the LPI cells, then all of a sudden you get a very strong response. And so the hypothesis or the idea was then that really the function of these inhibitory interneurons is to improve the flow field selectivity of the tangential cells. That's the function of these cells. And without them, they become very unselective. Now, going back to the final scheme, this is how we sort of imagined it before, asking, about these hypothetical inhibitory twins. They don't exist. What's going on is that each T4 and T5 cell makes synapses onto these inhibitory interneurons, which then take the signal to the adjacent layer of the lobular plate. This is how it's built. And with this, I want to close. I want to thank the Max Planck Society for supporting this research over so many years. I want to thank my colleagues at Genelia Farm, Gerald Rubin and Alyosha Nern, which provided all the driver lines. And without these driver lines, none of these experiments would have been possible here. I also want to thank the fly connectome team at Genelia. They did an incredible job with providing us with all the connectivity data. You don't have to go through 100 different candidate cell types. You can just pick the ones that are where they are connected to. This is tremendous work. But most importantly, I want to thank all the motion detectives at our department, which have really a diverse background. Some of them from molecular biology, others from physiology, others from computer science. And they all share sort of my enthusiasm of figuring out the neural implementation of the Reichardt detector. Thanks a lot.